Students, we are discussing chapter mineral nutrition. In mineral nutrition we completed a nitrogen cycle. Concerned to nitrogen cycle, I am writing some important statements. Okay, note. Number 1, listen this statement. Sir, denitrification. Due to denitrification process, nitrogen is moving from where to where? From soil to atmosphere. It means denitrification decreases the soil fertility in terms of nitrogen. Sir, nitrogen cycle, first step, nitrogen fixation, movement of nitrogen from atmosphere to soil, increases the soil fertility in terms of nitrogen. Yes or no? Whereas the last step of nitrogen cycle, denitrification. Okay, here nitrogen is moving from soil to atmosphere, therefore decreases the soil fertility in terms of what? Nitrogen. What it is? Denitrification. Therefore denitrification, what it is? Denitrification. What it does? Decreases soil fertility. Decreases what, sir? Soil fertility. Decreases soil fertility in terms of what? In terms of nitrogen. Okay, therefore denitrification increasing or decreasing the soil fertility? It decreases the soil fertility. Okay, right? Next, number 2. Sir, during biological nitrogen fixation, the first formed stable product is ammonia. During biological nitrogen fixation, the first formed stable product is what, sir? Ammonia. Right? During biological what, sir? Biological nitrogen fixation. During biological nitrogen fixation, first formed what? First formed stable compound is what, sir? Ammonia. First formed stable compound is ammonia. It's important. Okay, what is the first formed stable compound during biological nitrogen fixation? Ammonia. Does it mean that during biological nitrogen fixation, the first formed compound is ammonia? No. Yesterday, in the previous sessions, I showed you one diagram from the textbook. If you see the diagram, in that diagram, first what happened, sir? This nitrogen has formed N2H2. Then this N2H2 has formed what, sir? N2H4. What it is, sir? N2H4. Then it has formed what, sir? 2 molecules of ammonia. Yes or no? Right. Sir, what is this N2H2, sir? It is a diamide. What it is, sir? It's diamide, but it's unstable. Then what is formed? N2H4 is formed. This is called as a hydrazine. What it is called, sir? Hydrazine. It is called as a hydrazine. It is also unstable. Then finally what we got? We got 2 molecules of ammonia, which is stable. I think you got my point. Okay, right? Therefore, during biological nitrogen fixation, first formed stable compound is what, sir? Ammonia. First formed stable compound is what, sir? Ammonia. Right? Next, number 3. Sir, atmosphere contains a number of nitrogen oxides. Atmosphere is with number of nitrogen oxides. What is the source of this nitrogen oxides present in the atmosphere? Source of nitrogen oxides present in atmosphere. Source of what, sir? Nitrogen oxides present. Source of nitrogen oxides present in atmosphere. Present in what, sir? Atmosphere. We are discussing the source of nitrogen oxides present in atmosphere. Okay, first one is industrial combustion. What is the first one, sir? Industrial combustion. What it is? Industrial combustion. Second one is forest fire. What is the second one, sir? Forest fire. Third one is automobile exhaust. Automobile what? Exhaust. Automobile exhaust. And fourth one is power generating stations. Power generating stations. See the statement once. Sir, due to industrial combustion, forest fire, automobile exhaust, and in power generating stations, right, what are released? Nitrogen oxides are released into the atmosphere. Therefore, all these are sources of nitrogen oxides present in what, sir? Atmosphere, right? Now see this cycle once. Sir, this is a cycle for what, sir? Nitrogen, right? This is a nitrogen cycle. This is nitrogen cycle. Okay, sir, in this nitrogen cycle there are three main pools. There are three main pools for nitrogen. Three main nitrogen pools are: number one, atmosphere. First one is atmosphere. Second one is a soil. What is the second one, sir? Soil. Second one is a soil. And third one is a biomass. What, sir? Plant biomass, animal biomass, and a decaying biomass. Okay. Therefore, the three main nitrogen pools are atmosphere, right? Second one is a soil, and third one is what, sir? Biomass. What is the third one, sir? Biomass. Is this statements okay? Right. This concept is important. You should be careful with this flow chart of nitrogen cycle, right? Go for the next topic in mineral nutrition, right? Formation of root nodules in legume plants. What, sir? Formation of root nodules in legume plants. How root nodules are formed in legume plants? Okay, right? For example, right, this is root hair. This is what, sir? Root hair of what? Legume plants. This is the root hair of what, sir? Legume plants. Sir, in this right root hair, this white layer is a cell wall of root hair. This is what? Cell wall of root hair. Sir, this is plasma membrane of the root hair. This is what? Plasma membrane of the root hair. Plasma membrane of the root hair. What is this one? This is a epidermis. What is this one? Epidermis. Sir, I am taking a legume plant. Okay, legume means Fabaceae, dicot. Yes or no? Sir, what is this one, sir? Cortex. What is this one? Cortex. This is cortex. After cortex, what will come, sir? After cortex, you will get a steel. After cortex, what you will get, sir? Steel. Sir, in steel, what are present? First comes pericycle. First comes what, sir? Pericycle. Then comes vesicular bundles, that is a xylem and phloem. Right. Then comes medulla. Yes or no? Right. Okay. Right. Sir, take a legume root, Fabaceae family root. Okay. And cut it transversely. What you are going to see? Epidermis, cortex, and steel. Yes or no? In epidermis, you are going to see root hairs. For example, this is a root hair. In this root hair, this white layer is a cell wall of the root hair, whereas this violet one, this indicates a plasma membrane of the root hair. Plasma membrane of the root hair. Is this statements okay? Now listen. Sir, this roots of what? These roots of legume plants, they secretes sugars, amino acids, flavonoids into the soil. Sugars, amino acids, organic acids, flavonoids into the soil. Therefore, this root hairs of which plants are? Legume plants. It has secreted what, sir? Sugars, right? Amino acids, organic acids, and flavonoids into the soil. In response to these chemicals secreted by the root hair, Rhizobium bacteria gets attracted and moves towards what? Root hairs of legume plants. Is this statement okay? Right. Therefore, in response to the chemicals secreted by in response to the chemicals secreted by the root hairs of legume plants, Rhizobium bacteria is attracted towards the root hair of legume plants. It means the movement of this Rhizobium bacteria is in response to chemicals. So, movement of Rhizobium bacteria here is a chemotactic. What it is, sir? Chemotactic. It's chemotactic. Okay, right. Now listen, sir, all these are which bacteria are now? Rhizobium bacteria. All these are Rhizobium bacteria. Okay, right. Sir, this Rhizobium bacteria, which bacteria, sir? Rhizobium bacteria recognizes this as a root of legume plant. How, sir? With the help of ricadhesin present in Rhizobium bacteria. With the help of ricadhesin present in Rhizobium bacteria, right? Rhizobium bacteria is recognizing this as what, sir? Legume root. Is this statement okay? First I am writing these statements what I told just now. Okay, right? Therefore, root hair, root hairs of what? Legume plants secretes what? Secretes sugars, amino acids, organic acids, and what? Flavonoids were into the soil. Sir, what are flavonoids? In first year we read these flavonoids are secondary metabolites. Yes or no? They are secondary metabolites. They are secondary metabolites. Sir, in response to these chemicals, what, sir? In response to these chemicals, who secreted these chemicals? Root of legume plant. Yes or no? In response to these chemicals, Rhizobium bacteria is attracted. Rhizobium bacteria is attracted towards what? Towards roots of legume plants. Okay, therefore here Rhizobium bacteria is attracted, okay, in response to what, sir? Chemicals. Okay, so movement of Rhizobium bacteria is chemotactic. Yes or no? So here what? Rhizobium bacteria. Here Rhizobium bacteria shows which movement, sir? Chemotactic movements. Shows which movements, sir? Chemotactic movement. Shows chemotactic movement. Okay, right? Now once Rhizobium bacteria comes to the root of legume plants, the Rhizobium bacteria has to recognize whether it is a root of legume plant or not with the help of what? Ricadhesin protein. Okay, therefore with the help of what, sir? With the help of ricadhesin, with the help of ricadhesin protein, where it is present? Present in Rhizobium bacteria. Present in what, sir? Rhizobium bacteria. Present in Rhizobium bacteria. Rhizobium bacteria recognizes what? Recognizes Rhizobium bacteria. Rhizobium bacteria recognizes what? Legume roots. Is it okay? Right? Therefore, Rhizobium bacteria, it has recognized the right, Rhizobium bacteria, it has recognized the legume roots with the help of what, sir? Ricadhesin protein, which is present in Rhizobium bacteria. Once this recognition takes place, the Rhizobium bacteria is attached to the root hair and other epidermal cells of the root. Therefore, Rhizobium bacteria gets attached to the root hairs and other epidermal cells of the root. Other epidermal cells of the root. Is this statement okay? Right? Therefore, what happens, sir? Rhizobium bacteria. What? Rhizobium bacteria gets attached to what? Gets attached to root hairs and other epidermal cells, of root. What gets attached, sir? Rhizobium bacteria has attached to what, sir? To the root hair and other epidermal cells of the root. Okay, now what this Rhizobium bacteria does means it produces a complex polysaccharide. By name curling factor. By name what, sir? Curling factor. By name curling factor. Who produced this curling factor? Rhizobium bacteria. What is this curling factor? It is a complex polysaccharide. What this curling factor will do? It will curl the root hair. See the diagram. Sir, this is the actual root hair, but now the root hair has curled. Why the root hair has curled, sir? Due to the secretion of curling factor. Who secreted this curling factor? Rhizobium bacteria. What is this curling factor? It is a complex polysaccharide. Therefore, Rhizobium bacteria secretes a curling factor. Curling factor is a complex polysaccharide. This curling factor will curls the what? Root hair. Now this curled root hair is called as shepherd's crook. What is this curled root hair called as a shepherd's crook? What it is called as? Shepherd's crook. Curled root hair is called as what, sir? Shepherd's crook. Sir, how this curling factor was recognized by the root, sir? This legume root contains a protein by name lectin. Lectin protein. Lectin is also a secondary metabolite. In first year we got it. Lectin, secondary metabolite. Yes or no? Right. With the help of this lectin present in the legume root recognizes what's our curling factor. Is it okay? Right? Right. Now, which bacteria that is? Rhizobium bacteria. Rhizobium bacteria. Sir, what it do, sir? Rhizobium bacteria produces a what? Curling factor. Produces a curling factor, which what it does? Which curls what? A root hair of which plants? Legume plants. Sir, what is this curling factor, sir? This curling factor is a complex polysaccharide. Is a complex polysaccharide. Okay, which is going to curl the what's our root hair of legume plants. Okay, right? This curled root hair. What's sir? Curled root hair. What is this curled root hair called as, sir? Called as a right. What's sir? Shepherd's crook. Right? What it is? Shepherd's crook. Called as a shepherd's crook. Okay, what is this curled root hair called as a shepherd's crook? Now, sir, this curling factor, right, recognized by legume roots with the help of what, sir? With the help of lectin protein. With the help of what, sir? Lectin protein. This lectin is also a secondary metabolite. It is also a secondary metabolite. Where this lectin protein is present, which is present in legume root, which is present in legume root. Okay, now see this diagram, sir. Once the root hair of legume plant gets curled, then what happens means this Rhizobium bacteria produces cell wall dissolving enzymes. Produces what, sir? Cell wall dissolving enzymes. Okay, cell wall dissolving enzymes or cell wall digesting enzymes. Who is producing the cell wall digesting enzymes are Rhizobium bacteria. These cell wall digesting enzymes are cell wall dissolving enzymes. They dissolve the cell wall of root hair. What gets dissolved? The cell wall of root hair gets dissolved. So the plasma membrane of the root hair gets exposed. It means now it is exposed to Rhizobium bacteria. What gets exposed to Rhizobium bacteria, sir? Plasma membrane of what? Root hair. When the cell wall of the root hair gets dissolved, how? With the help of cell wall dissolving enzymes. Who secreted them? Rhizobium bacteria. Therefore, Rhizobium bacteria secretes cell wall dissolving enzymes, by which the cell wall of root hair gets dissolved. So plasma membrane gets exposed to Rhizobium bacteria. Now the Rhizobium bacteria enters into the plasma membrane of the root hair. Rhizobium bacteria enters into the plasma membrane of what? Root hair. Once Rhizobium bacteria enters into the plasma membrane of the root hair, this plasma membrane of the root hair invaginates into the deeper layers of the root in the form of a thread. This thread is called as infection thread. What is this thread called as? Infection thread. Why, sir? As it carries Rhizobium bacteria into the deeper layers of the root. Okay, sir, once Rhizobium bacteria enters into the plasma membrane of root hairs, the plasma membrane of root hair has invaginated into the deeper layers of the root in the form of a infection thread. This infection thread, what it carries, sir? Rhizobium bacteria. Carries Rhizobium bacteria. When this Rhizobium bacteria, they are moving through the infection thread into the deeper layers of the root, Rhizobium bacteria goes on dividing and increasing its number. Who? Rhizobium bacteria, right? When they are moving through the infection thread, Rhizobium bacteria goes on dividing and increasing its number. Right? Right. Therefore, what happened here, sir? Sir, shepherd's crook is formed. Okay. Now, Rhizobium bacteria, what it is doing, sir? Rhizobium bacteria secretes what, sir? Cell wall dissolving enzymes. Okay, by which what happens? Cell wall of root hair gets dissolved. What gets dissolved, sir? Cell wall of root hair gets dissolved with the help of cell wall dissolving enzyme secreted by Rhizobium bacteria. So plasma membrane of what? Root hair gets exposed. Plasma membrane of root hair gets exposed. Now what happens? Rhizobium bacteria enters into what, sir? Plasma membrane of root hair, by which then what happens? Root hair, okay, plasma membrane of root hair Plasma membrane of root hair. Exvaginates, invaginates, invaginates. Invaginates into where? Deeper layers of root as in the form of what? As a infection thread. Sir, what this infection thread carries? Carries Rhizobium bacteria, right? This infection thread carries Rhizobium bacteria. This infection thread carries what it carries? Rhizobium bacteria. Okay, right? As Rhizobium bacteria, right, moves through what? Infection thread. Infection thread, what happens? Bacteria divides. Bacteria divides. Okay. Now listen this statement. Sir, this is a Rhizobium bacteria. Okay. Sir, now this Rhizobium bacteria, what it does means they secretes a mitogen. Rhizobium bacteria secretes a mitogen. Mitogen means which helps in mitosis. In response to this mitogen, right, the legume plant also secretes a stimulant. With the help of this mitogen and the stimulant, okay, the cortical cells, which cortical cells? Inner cortical cells and pericycle cells of legume plant starts dividing and produces a structure called as a nodule. Produces a structure called as what? Nodule. Produces a structure called as nodule. Sir, Rhizobium bacteria is inside the infection thread. Rhizobium bacteria is inside the infection thread. This Rhizobium bacteria secretes a mitogen. In response to this mitogen, the legume root also secretes a stimulant. With the help of this mitogen and stimulant, what happens, sir? What happens, right? The inner cortical cells and pericycle cells of legume plant divides and produces a nodule. It means inner cortical cells and pericycle cells are involved in the formation of root nodules in legume plants. What and what? Inner cortical cells and pericycle cells are involved in the formation of what, sir? Involved in the formation of root nodules in legume plants, right? Okay, right? Now what happens? Rhizobium bacteria. Rhizobium, what? Bacteria. Rhizobium bacteria, right? Rhizobium bacteria, what it do, sir? Produces. Rhizobium bacteria produces a what? Mitogen, means which helps in mitosis. Okay. In turn, host legume plant produces a what's a stimulant. Host legume plant has produced a stimulant. This mitogen and stimulants, right, stimulates what? Inner cortical cells and pericycle cells of legume root to divide and produce a nodule. Therefore, in the formation of root nodule, in the formation of what? Root nodule. In the formation of what, sir? Root nodule. In the formation of root nodule, right, inner cortical cells inner cortical cells, and what more, pericycle cells of legume root are involved. Who are involved in the formation of a root nodule in legume plants, sir? There is inner cortical cells and pericycle cells of legume roots. They are involved in the formation of root nodule. Okay. Now see this statement, sir. For example, right? This is a cortical cell, sir. With the help of this stimulation, the cortical cells divided. What gets divided? Cortical cells gets divided. When these cortical cells and pericycle cells gets divided, what happens? A nodule is formed. Sir, the cells in this nodule, they are polyploid. Sir, polyploid, generally tetraploid. Okay. Now see here, sir, for example, right, this is the infection thread, sir. What is this one, sir? Infection thread. What it is carrying, sir? Rhizobium bacteria. Now what happens means, slowly, right, this infection thread, what, sir? This infection thread starts budding. Budding, right? Due to this budding, what happens means small vesicles are formed. What are formed, sir? Small vesicles are formed. Each vesicle contains either one Rhizobium bacteria or more than one Rhizobium bacteria. See this statement once, sir. This is infection thread, sir, carrying what, sir? Rhizobium bacteria. What is happening? Slowly this infection thread, it is producing buds. That bud gets separated from the rest of the infection thread and behaves as a vesicle. What that vesicle carries? Either one or more than one Rhizobium bacteria. Once this Rhizobium bacteria is inside the vesicle, the bacteria stops dividing and nitrogenase enzyme present in this bacteria gets activated and starts fixing the nitrogen from atmosphere. Okay, this group of Rhizobium bacteria, okay, called as a bacterioid with active nitrogenase enzyme. With which enzymes are? Active nitrogenase enzyme, right? Now what is happening, sir? Infection thread starts budding and produces a what? Small vesicles. Okay, each vesicle either with one or more than one Rhizobium bacteria. Once inside the vesicle, what? Vesicles, what happens? Rhizobium bacteria stops dividing and nitrogenase enzyme gets activated. What gets activated? Nitrogenase enzyme gets activated. This group of Rhizobium bacteria with active what? Nitrogenase enzyme, and surrounded by peribacterioid membrane. Peribacterioid membrane. This group of Rhizobium bacteria with active nitrogenase enzyme, and this group surrounded by what, sir? Peribacterioid membrane called is called what, sir? Bacterioid. What it is called as? Bacterioid. Is called as what, sir? Bacterioid. See here, sir, all these are Rhizobium bacteria. This Rhizobium bacteria, right, they are surrounded by what, sir? Peribacterioid membrane. This peribacterioid membrane is nothing but what? Plasma membrane. Plasma membrane of what, sir? Host cell, that is a legume plant. Therefore, this peribacterioid membrane belongs to what, sir? Host plant. Host plant. Okay, now Rhizobium bacteria makes direct contact. Rhizobium bacteria makes direct contact with vascular tissues of the host plant for exchange of nutrients. Rhizobium bacteria, right, present in the root nodules makes a direct contact with the vascular tissues of the host plant for exchange of nutrients. Rhizobium bacteria present in the root nodules of legume plants makes a direct contact with the vascular tissues of the host plant for exchange of nutrients. Okay, right, sir, in root nodules of legume plants the condition is polyploid, generally tetraploid, right? Thank you.